Welcome back to the deep dive. Today we are strapping ourselves into the cellular submarine and diving deep into the microscopic machines that make life quite literally possible.
We really are.
We are talking about enzymes, the fast, efficient, and surprisingly complex engines that power every living system.
These are arguably the most crucial class of molecules in biology. I mean, they're just background players. They're the specialized biological catalysts that govern nearly every single metabolic reaction in your body.
So if they stall or break.
Life stops. It's that simple.
The mission for this deep dive is to start with their fundamental power, how they manage to generate all that speed at such gentle temperatures, then explore the delicate environmental controls that keep them functional.
And then the really cool part.
And finally, yes, we'll cap it off by seeing the sophisticated math scientists use to actually track these invisible engines, moving from, classical averages to the unpredictable reality of molecular randomness.
It's a fantastic journey.
Okay, so let's unpack the core idea. Our sources define enzymes as large protein molecules that act as catalysts. We all know a catalyst speeds up a reaction without being consumed, but that definition, it really undersells the scale of the speed.
It absolutely does. The real insight here is the magnitude. Without the enzyme, a necessary reaction might take, billions of years to complete on its own?
Billions of years, right.
With the enzyme, it takes a fraction of a second. They provide a route for chemical transformation that is perhaps a billion times faster than the uncatalyzed reaction. That insane jump in speed is the definition of life.
And their superpower, the core mechanism that allows this, is lowering the energy of activation.
Right, so think of every chemical reaction needing to get over a hill to proceed. That hill is the activation energy barrier.
Okay.
In a chemistry lab, you might add tremendous heat or pressure to force molecules over that barrier. But inside you, adding that kind of heat, it would damage or destroy cells.
It would cook you.
Exactly. So enzymes provide an alternative lower path through the hill, allowing the reaction to proceed swiftly and efficiently at the gentle, life-sustaining temperatures found in the human body, typically around 37 degrees Celsius.
Now, let's quickly define the structural players involved. We have the substrate,
which is the starting material, the thing you're working on.
And the product, the end result. But the most critical part is the active site.
That's the business end. The active site is a specific three-dimensional groove or pocket on the enzyme protein where the substrate physically binds.
And where the magic happens.
And where all the catalysis takes place, yes. We emphasize that it's three-dimensional because its precise shape determines the entire function of that protein. Change the shape, you lose the function.
And that links directly to the concept of specificity. Our sources stress that enzymes are almost always molecular specialists.
Oh, absolutely. Generally, one enzyme speeds up one reaction only, or at most, a very small group of related reactions.
And this specificity is vital for maintaining metabolic order, isn't it? If one enzyme could do 100 different things, it would be.
It would be chaos.
Yeah.
Total cellular chaos. Because they are so specific, we can build these finely tuned, incredibly complex metabolic pathways.
And we see the immediate importance of this specialization everywhere, from, say, construction to demolition.
Right. Think about the breakdown crew inside your gut. We need specialized enzymes to tackle specific food groups. Amylase only works on starch.
Lipase for fats.
And protease only works on proteins. They are targeted demolition experts.
But they're also the detox team. I mean, if you've ever poured hydrogen peroxide on a cut and seen that furious froth.
You're watching an enzyme in action.
Yeah.
That bubbling is the enzyme catalase, which is one of the fastest enzymes known. It immediately breaks down poisonous hydrogen peroxide, a common cellular waste product, into harmless water and oxygen. That immediate and specific action is key to cellular survival.
And they don't just break things down.
No, not at all. Other specialized enzymes, like starch phosphorylase in plants, are construction crews, building large, complex molecules like starch from smaller glucose units.
So moving beyond the players, let's look at how the substrate actually fits into that active site. We have two historical models. The older one, dating back to the 1890s, is the lock and key model.
Right, and that historical view envisioned the active site as a rigid, unmoving lock. Only one substrate, the key, would fit perfectly into it, forming an enzyme-substrate complex.
It was elegant and simple.
Very, but not entirely accurate.
Because the more accurate picture today is the induced fit model. This suggests the enzyme is actually a flexible, dynamic structure. It's less like a rigid keyhole and more like a high-tech glove.
Exactly. When the substrate approaches, the active site doesn't just passively accept it. It actually reshapes slightly, tightening around the substrate to create a closer, more strained fit.
Then that strain is the important part.
That induced stress is critical because it forces the substrate's bonds into a transition state, making the chemical reaction much, much easier. The enzyme is actively participating, not just passively waiting.
So regardless of how it binds, the result is the same, the catalytic cycle. The enzyme and substrate meet, the reaction occurs, the product is released.
And the enzyme immediately springs back to its original shape, ready to repeat the process.
Ah.
They're incredibly reusable, catalyzing thousands of reactions every second.
But the fit might be perfect, but that perfection is fragile. What external forces can threaten this incredible efficiency?
Well, that's the next big question.
Exactly. These tiny machines are extremely sensitive to their environment, and we have two primary factors, temperature and pH. Let's start with heat.
Okay. Within a certain range, increasing temperature increases the rate of reaction. It just makes sense. The kinetic energy of the molecules increases, leading to more frequent collisions between the enzyme and the substrate.
So more collisions, more reactions.
Right, and this boosts the reaction rate up to an optimal point for human enzymes. That sweet spot is right around 35 to 37 degrees Celsius.
But push it too far, and you get a catastrophic failure known as denaturation.
Yeah. If you push the temperature typically above 40 or 50 degrees Celsius, the entire protein structure begins to vibrate violently. Since the enzyme's a protein, that high heat disrupts the delicate hydrogen and ionic bonds that maintain its three-dimensional shape.
It just falls apart.
It essentially unravels. And once that active site deforms, it can no longer fit the substrate, causing the activity to drop sharply, often permanently. It's no longer a perfectly fitted glove, it's a melted blob.
And the other major environmental factor is pH. How acidic or alkaline the surrounding solution is makes a massive difference.
And it connects directly to the enzyme's molecular structure. See, pH is crucial because it alters the ionization of the R groups, the chemical side chains on the enzyme's amino acids.
So those little side chains determine the folding.
They determine everything. So changing the ionization fundamentally changes the enzyme's electrical properties, and thus its shape.
And we see striking contrasts based on where the enzyme operates in the body. Take pepsin.
Which works in the highly acidic environment of the stomach. Its optimum performance is at pH 1.5 to 2.0. I mean, that's battery acid territory.
Wow. Contrast that with trypsin, which works in the small intestine where things are neutral to slightly alkaline.
And its optimum pH is way up around 8.0 to 9.0. If you swamp them, if you put pepsin in the small intestines environment, it just stops working.
Instantly.
Instantly, its delicate shape is immediately disrupted. Extreme pH, just like extreme heat, is a major denaturing agent.
Finally, we need to consider concentration. The reaction rate increases linearly with both enzyme and substrate concentration until you hit a wall.
Right, and that wall is reached when the enzyme hits its maximum velocity, or Vmax. This happens when the substrate concentration is so high that virtually all active sites on all the available enzymes are continuously occupied.
A state we call saturation.
Saturation. And this is where regulation becomes critical. If the cell reaches Vmax consistently, that implies a massive waste of resources. The cell is producing far more product than it needs, and it can't increase the speed anymore.
So how does the cell prevent that continuous saturation and maintain efficiency?
Well, it relies on sophisticated regulatory switches.
They don't just run constantly.
No, not at all. They are controlled by other molecules, chiefly inhibitors, which slow down or stop activity, or activators, which kick them into gear.
Let's focus on inhibitors because they illustrate the fine control. We have two main types. First, constitutive inhibitors. These are molecular imposters.
They're structurally similar to the actual substrate, and they compete directly for the active site, like someone parking illegally in a designated parking spot.
That's a great analogy.
Yeah, the antibiotic penicillin, for example, is a competitive inhibitor. It blocks a crucial enzyme needed by bacteria to build their cell walls. If the imposter is there, the reaction stalls.
Then you have the fascinating non-competitive or allosteric inhibitors. They don't go anywhere near the active site at all.
Correct. They bind to a completely different location on the enzyme, called the allosteric site. Binding there acts like changing the ignition key on a car. It causes a distant change in the enzyme's overall conformation.
Which warps the active site's shape.
And makes it useless for the substrate. This is a much more powerful regulatory mechanism because no matter how much substrate you pour in, the reaction won't happen if that allosteric site is blocked.
And speaking of fine control, we need to talk about feedback inhibition. This isn't just a switch, it's a thermostat for metabolic production.
This is one of the most elegant pieces of cellular control. So imagine a smart manufacturing assembly line designed to produce, let's say, molecule Z.
Okay.
Instead of waiting for a supervisor to check stock, the final product, molecule Z itself, loops back and acts as an allosteric inhibitor on the very first enzyme in that pathway.
So if there's a sufficient supply of Z already built up, Z itself hits the off switch on the start of the entire production line.
Exactly. When the concentration of the final product falls again because the cell is consuming it, the inhibitor detaches, the first enzyme regains its shape, and the assembly line kicks back on.
It's an instantaneous self-regulating mechanism.
Prevents resource waste and unnecessary buildup. It's beautiful.
This raises a really important question. If these reactions are so crucial and so finely regulated, how do scientists actually model these reaction rates in a way that captures both the average speed and the tiny, unpredictable details?
This is where we see a real intellectual divide between the classical approach and the modern computational view.
Let's start with the classic. That's the deterministic model, dominated by the Michaelis-Menten equation, right? It's been around for over a century.
It has. This classical approach uses ordinary differential equations, or ODEs. You can think of ODEs as the tools of calculus used to calculate smooth, continuous, predictable change.
Okay, so it assumes a big system.
A huge system. It assumes a large, well-mixed reaction environment with high molecule counts, thousands or more. And it treats the whole process as a continuous flow, successfully predicting the smooth average rate of reaction.
And to make the math work, they rely on something called the quasi-steady-state assumption, or QSSA.
That assumption is key. It basically assumes that the concentration of the enzyme-substrate complex forms very quickly and then remains constant or at a steady state during the main part of the reaction.
So it smooths things out.
It mathematically smooths out any rapid initial fluctuations, allowing them to calculate that smooth average rate. This works perfectly if you're looking at the overall freeway speed of metabolism.
But that perfect, smooth predictability vanishes when you start looking at the individual car, or in this case, the individual molecule inside a single cell.
Exactly. What's fascinating here is that when molecule counts are small tens or hundreds, which is very common in cellular contexts, random fluctuations, what kineticists call molecular noise, become non-negligible. The deterministic model simply can't capture that randomness.
So if the classical view gives us the average freeway speed, what gives us the unpredictable speed of every single car on that freeway?
For that, you need stochastic models. These probabilistic models, using methods like the Gillespie algorithm, treat every single reaction event probabilistically.
So it's not a smooth curve anymore.
No. Instead of predicting a smooth curve, they track the time evolution of the probability of having specific integer counts of molecules. They acknowledge that two identical cells running the exact same reaction might yield wildly different results over a short period. That is the definition of dynamic disorder.
But it's amazing that when scientists conduct single enzyme turnover experiments in the lab, they confirm that while stochastic fluctuations cause this wide distribution of waiting times between products,
The mean rate
still conforms to Michaelis-Menten kinetics.
It's the perfect harmony between the two views. The deterministic result, the smooth, predictable average, is simply what emerges when molecule counts become large. It's a beautiful illustration of the law of large numbers applied to biochemistry.
So the stochastic model is really the foundation.
It serves as the foundational knowledge, showing that the cellular world, at its smallest scale, is ultimately probabilistic.
It's incredible that our understanding of enzyme action requires two such mathematically opposite perspectives, depending entirely on the scale you are examining.
And because of that incredible precise efficiency, we utilize enzymes commercially everywhere, far outside the cell walls.
Absolutely. Enzymes are essential in the food industry. We use amylase for pre-digesting baby food and specialized enzymes for softening chocolate centers.
And perhaps the most common industrial use is in cleaning. The proteases in biological washing powders are amazing.
How do they work?
They break down protein-based grime and dirt large insoluble molecules into smaller soluble molecules that can be easily washed away. Crucially, their thermal stability allows the powder to work effectively at lower temperatures.
Which saves a huge amount of household energy.
And a massive amount.
However, industrial use isn't without risk for the people working directly with them in manufacturing settings, like pharmaceuticals or detergents.
That's right. Because enzymes are proteins, chronic occupational exposure carries a significant health risk. Studies show a surprising number between 12 and 27% of enzyme manufacturing workers become sensitized,
Sensitized, leading to.
often leading to allergic disease and work-related asthma.
That's why monitoring is so crucial then. Air sampling using specialized cassettes is required to measure exposure levels.
And while there are currently no official U.S. government limits for enzyme exposure, the accepted reference exposure limit for common proteins like sudelacin is set very low, 60 nanograms per cubic meter of air.
60 nanograms, that's incredibly small.
It is. It just illustrates how potent these tiny molecules are, even outside the cell.
That brings us to the end of our dive. We've traveled from the specific, flexible fit of the induced fit model, learned how temperature and pH create a fragile yet essential environment for these catalysts,
And explored the complex mathematics required to model them, from the smooth average to the random event.
We've learned that the life-sustaining average speed of enzyme turnover is accurately predicted by classical deterministic math. But the modern view, supported by single enzyme experiments, reveals that at the ultra-small scale inside a cell, the reaction rate isn't a steady, predictable curve.
But a series of probabilistic, fluctuating events. The inherent noise, the dynamic disorder, is always present.
So let me leave you with this final provocative thought for you to consider. If the fundamental biochemical processes keeping you alive rely on random molecular collisions, how much of metabolic control relies not just on perfect on-off switches like feedback inhibition, but on the capacity of the system to simply manage and buffer inevitable chaos? What might happen when a key enzyme's copy number drops from 100 molecules to just 10? At that incredibly small scale, the inherent noise ceases to be a background hum and instead becomes the defining feature of life's precise yet ultimately random molecular engines.